Welcome back. In our final video for Chapter 5, we'll be looking at algae and lichens. And by the end of this section, this video, you should be able to 1. Explain why algae are part of microbiology, 2. Describe their unique characteristics, 3. Identify toxin-producing algae, 4. Compare the major algal groups, and 5. Classify algae into their major supergroups. So why study algae and microbiology? Algae are autotrophic protists. That means they create their own food using basic energy sources like sunlight or chemicals rather than eating other living things. So they're also known as producers that can convert inorganic materials like carbon dioxide and water into organic compounds like glucose in order to fuel their own growth and energy. They can be unicellular like plankton or multicellular like seaweeds. They also produce about 70% of the Earth's oxygen through aquatic photosynthesis. They are important in food chains and are used in labs and food production as well. They have these rigid cell walls containing agar or carrageenan, which are really useful as food thickeners or solidifying agents in cosmetics, toothpastes, or even foods like mochi. And they can also be used as a solidifier to add to growth media for our microbes. And we've seen these in the lab so far in our test tube slants. Soon we'll see them in our petri dishes and our plates in lab as well. But not all algae are beneficial. Some produce toxins, which can lead to harmful algal blooms. Algae are eukaryotic and photosynthetic. They contain chloroplasts for photosynthesis, some from primary endosymbiosis and others from secondary. Primary chloroplasts have 2 membranes, one from the original cyanobacterium that was engulfed, and one from the plasma membrane, so this outer plasma membrane of the engulfing cell. So I can see 2 membranes here, the inner darker green membrane and the outer darker yellow membrane. In secondary endosymbiosis, we have chloroplasts with multiple membranes, and that is because what's happening is this organism is being engulfed by another eukaryotic cell. So ultimately we add additional membranes such as that membrane, as well as the membrane of the new engulfing cell in blue. Different pigments within the various types of algae can produce red, brown, or green varieties of algae. Unlike plants, algae lack true roots, stems, or leaves, or even a waxy cuticle, which is a protective outer layer that's water repellent that we find on the surface of most plants. Finally, algae are kind of like plants in that they can reproduce both sexually or asexually. If we look at the organization of algae and algal diversity, they are organized into supergroups. One used to be called Chrome alveolata, but that's outdated. The groups once lumped into Chrome alveolata are now placed under the SAR supergroup. These are the Straminophiles, alveolates, and Rhizaria supergroup. Although you might still see Chrome alveolata used in some of the older references out there. So today these fall under the SAR supergroup: dinoflagellates, diatoms, golden algae, and brown algae. And the second big group is, or are, the Archaeplastida, and this includes green algae, red algae, charophytes, and land plants. Dinoflagellates are our first algal group within the SAR supergroup, and they're mostly marine plankton. Remember that plankton are diverse aquatic organisms that kind of drift with our ocean currents. For dinoflagellates, they have 2 flagella, which give them a spinning movement. Usually they have one wrapped around like a belt, and then they have one here at the end of the cell. Some of them are also armored with cellulose plates, like the one shown here. They look like a hard shell covering the dinoflagellate. And certain species can produce neurotoxins that cause paralytic shellfish poisoning. So examples of dinoflagellates that can make neurotoxins include Gonulix and Alexandrium. When they bloom densely, these dinoflagellates can cause something known as a red tide. And it's called a red tide because dinoflagellates can contain a variety of pigments. Some of them contain red pigment, brown, or reddish pigments. And when these organisms multiply in really high numbers, the water will take on some kind of a reddish or brownish color. But not all blooms are red. So sometimes they can be green, brown, or even colorless, and that'll still be called a red tide. So a better term to use is harmful algal bloom that you'll see throughout the slides as well. In going back to paralytic shellfish poisoning, shellfish can make you sick. It can cause a serious illness, and shellfish are things like mussels, clams, oysters, or scallops. They can make you sick if they've accumulated neurotoxins produced by these dinoflagellates during the harmful algal blooms or red tides. Within the stromatophiles, which are in the SAR supergroup, we have diatoms, golden algae, and brown algae. Diatoms have silica cell walls called frustules, and they're important in diatomaceous earth. So their fossilized remains can be used to produce diatomaceous earth, which has a range of uses such as filtration or insulation. And up here in the middle, we have a scanning electron microscopy image of the cell walls of different types of diatoms here. For golden algae, these contain pigments such as chlorophyll or fucoxanthin, which has kind of a brownish or yellow-brown carotenoid pigment. Some species can cause fish kills in freshwater, and this is when toxic blooms of golden algae release chemicals that suffocate fish by damaging their gills, leading to really huge or massive die-offs of these fish. And this is golden algae on the right. Finally, we have brown algae. Brown algae, like giant kelp, are multicellular, and they form these seaweed forests in marine environments. Our next supergroup are the Archaeplastida, and these include red and green algae. For green algae, we have two phyla, Chlorophyta and Charophyta. These all have chlorophyll A and B, cellulose in their walls, and they can store starch. In terms of their shape and morphology, they range from unicellular organisms like Chlamydomonas, shown here on the left, to colonial organisms like Volvox in the middle, to multicellular algae like Ulva, which is also sometimes known as sea lettuce. Then we also have red algae, or Rhodophyta. These are mostly multicellular, and they have rigid cell walls containing agar or carrageenan. These are both important for food and lab growth media. So red algae is commonly used in food, maybe as a seaweed salad, or in mochi as a thickener, and also as nori. If you eat sushi or something similar, nori is a type of dried, edible red algae. It's pressed into these really thin paper-like sheets, and it turns this dark, like blackish green color because the red pigment is pretty heat sensitive, and that processing method of shredding and drying the algae also contributes to this darker color. We also see it a lot in lab. So we said red algae has agar and carrageenan, and we can use agar in our growth media. So far we've seen slants. Pretty soon we're going to be working with these deep tubes as well as Petri plates. Agarose is derived from agar, which comes from our red algae as well. And agarose is used as a gelling agent and kind of a molecular sieve or filter in a lot of biology experiments, with the most common application probably being agarose gel electrophoresis. This is a technique that we use to separate DNA, RNA, or proteins by size. And this is a figure from our book that nicely shows us the diversity, all the different types of algae out there. We have brown algae here on the upper left. These are large multicellular kelps. In the middle, we have multicellular red algae. And on the right, we have a green alga that looks kind of like a plant, but it's not. These are algae still. In the middle row, we have some glowing blue. This is bioluminescence due to dinoflagellates. So some dinoflagellates have this light-emitting compound called luciferin, and the enzyme luciferase will catalyze the reaction, causing this light to be emitted. And this is assumed to be some kind of defensive mechanism that might startle their predators or maybe make them more visible to larger predators. On the right, we have diatoms. Remember, these have silica walls that form diatomaceous earth. And at the bottom, we have images of green algae known as volvox. So remember, these are colonial algae, and you might have seen these before in Bio-21 or in Majors Biology, where you would have to actually take your slide and put a few sand grains before you add the sample or before you add the cover slip to prevent destroying these three-dimensional spherical organisms. And this is just for fun, but if you enjoy kayaking, there's actually a place that you can go to see the bioluminescence of dinoflagellates that's relatively close to our area. So it is over here in Tamales Bay, so just north of San Francisco. So it varies, so you have to kind of search to see when the season begins, but sometime from late spring to early fall, there's usually a period of time when the dinoflagellates are really emitting that bioluminescence. And this is great if you have time or interest to do this. I would recommend it. I would recommend going kayaking first in the daytime if you've never done it. Practice a few times before you go at night, because that can be kind of scary if you've never done it before. So for our checkpoint here, the groups of algae most associated with harmful algal blooms are those dinoflagellates. And these were within the supergroup SAR, formerly known as Chrome alveolata. Dinoflagellates like Gonulix or Alexandrium can produce neurotoxins, and these neurotoxins can cause paralytic shellfish poisoning. So remember, when humans are exposed to contaminated water or contaminated shellfish from water with these harmful algal blooms, we can get sick. So these are caused by dinoflagellates during the red tides, and that's algae. In the past, in a previous video, when I was talking about bacteria, bacteria like cyanobacteria can also cause algal blooms, although those are not algae. So if I'm asking for algae, we want to say dinoflagellates. If I'm asking for bacteria, you can reference or say cyanobacteria. All right, let's recap section 5.4. So why are algae included in microbiology? Many algae are unicellular or microscopic, even though some of them are huge or macroscopic like seaweed. Overall, their structures, reproduction, and ecological roles are studied at the microbial level, and they're important in our micro labs because they're a source of agar, agarose, and carrageenan. For the unique characteristics of algae, we said that they are eukaryotic, they're autotrophic, so they're photosynthetic. They have chloroplasts. They produce around 70% of the Earth's oxygen in aquatic environments. We saw earlier that they lack true tissues, roots, stems, or leaves like plants do, or like plants have, although they did have a variety of pigments like red, brown, or green algae, and they could reproduce sexually and asexually. For the third, examples of toxin-producing algae, we talked about those dinoflagellates, like Gonulix or Alexandrium, that can cause paralytic shellfish poisoning, and they can cause that during the red tides. For the fourth, comparing major groups, so we had our SAR supergroup, or the formerly known Chrome alveolata. These included dinoflagellates that cause the red tides, the diatoms that have the silica cell walls used in diatomaceous earth, and we had brown algae that formed those giant kelp. For the second supergroup, we talked about Archaeplastida, which included green algae like the unicellular Chlamydomonas, the colonial volvox, or the multicellular ulva. And then we talked about red algae. These are mostly multicellular. They had cell walls with agar or carrageenan. So for classification according to those major groups, again, we have two supergroups we talked about. So the supergroups were the SAR supergroup, which included dinoflagellates, diatoms, golden algae, and brown algae. And the second supergroup was the Archaeplastida, which included green algae and red algae. It also includes charophytes and land plants, but we didn't really talk about that, so focus on just green and red algae in the second supergroup, Archaeplastida. And now we're going to move on to lichens in section 5.5. By the end of this section, you should be able to explain why lichens are studied in microbiology, describe their unique symbiotic relationship, and identify ways that they benefit our environments. So what are lichens? Lichens are actually a symbiotic partnership between a fungus and some kind of photosynthetic partner. And this can either be green alga or cyanobacterium. So they work together because the fungus provides structure, protection, and minerals for the photosynthetic partner, and the photosynthetic partner provides carbohydrates to the fungus through photosynthesis. Some scientists consider this symbiotic relationship to be actually a type of parasitism. Remember, parasitism is when one organism benefits but the other is harmed, because the photosynthetic organism actually grows less well than it would without the fungus. Although this relationship also allows the photosynthetic organism to grow outside of water, which is usually not possible for green algae or cyanobacteria. So whereas algae normally grow only in aquatic or extremely moist environments, lichens actually can be found on almost any surface, even rocks, or as epiphytes, which means that they can grow on other plants. Lichens grow on rocks then, or trees, soil, or even in extreme environments. They're important in soil formation because they can break down rock, and they're also very sensitive to pollution, which makes them great environmental indicators. So because lichen absorb water and nutrients directly from the atmosphere instead of through roots, they also accidentally absorb pollutants in the air. So many of them are extremely sensitive to air pollution. So if the air is polluted, they're going to stop growing or completely die and disappear. By looking at which lichens are present or absent in an area, then scientists can actually estimate air quality and pollution levels. So if you're going hiking and you see an area with a ton of lichen, you can be pretty sure that the air is pretty good there. It's pretty clean. But if you see those start disappearing, you start thinking, oh, something is lowering the air quality and there's more air pollution, leading to these loss of lichen. And for reproduction, lichen can reproduce through small fragments that contain both partners, the fungus and the photosynthetic partner, and they actually are pretty slow growing and can even live for centuries. For their growth forms, there are three main types. Crustose, which is shown here, is tightly attached to the surface and has that crusty appearance. Foliose, these have leaf-like flat sheets. And fruticose, shown on the right, these are branched and they have shrub-like structures. Lichens play critical ecological roles, so they're often the first colonizers in harsh environments. It turns out they're pretty hardy or robust in nature. They can tolerate physical extremes like heat, cold, drought, poor rock, or even poor soil, so they're great pioneer colonizers of harsh environments like deserts or volcanic rock or mountaintops. But they're really sensitive to pollution, like we mentioned earlier. So it turns out lichens are tough against natural stresses, but fragile against man-made pollution. Lichen can also serve as food for some animals like caribou. You can see some feeding over here. And environmentally, we mentioned that they're great bioindicators of air quality. Humans have also used them for dyes or perfumes or traditional medicine. For example, there's this type of lichen called oak moss that's used in fragrances, and some lichens actually have antimicrobial properties. For our lichens checkpoint, what kind of organisms are found in lichens? We have that fungus, which provides protection, structure, and minerals, and also some kind of photosynthetic partner. This can be green alga or cyanobacterium that provides carbohydrates through photosynthesis to the fungus. For the three growth forms, we talked about crustose, which is tightly attached to the surface and crust-like, foliose, which is flat and leaf-like, and fruticose, which is branched and shrub-like. All right, and now we can recap the entire section for 5.5 lichens. Why we study lichens in microbiology? Because lichens include fungi, algae, or cyanobacteria, all of which are studied in microbiology. The fungal and algal components are often microscopic, and we know that cyanobacteria are microscopic as well, and their symbiotic relationship really helps us understand microbial ecology. For the second one, unique characteristics, they are a composite organism made of at least two partners living together, so that's pretty rare. Their fungal partner, remember, provides that structure, protection, moisture, and it absorbs minerals for the photosynthetic partner. And in return, the photosynthetic partner, that green alga or cyanobacteria, provides carbohydrates through photosynthesis to the fungus. And for the third, different ways lichens are beneficial to the environment, so they can undergo soil formation. They can break down rock surfaces, contributing to new soil. They are often first colonizers. They can grow in these extreme environments where other organisms cannot. They are great environmental indicators. Since they're sensitive to pollution, we can use them to monitor air quality. And finally, they can provide food for some animals like caribou or different species of deer. And that takes us to the end of Chapter 5. In Chapter 6, we'll start exploring acellular pathogens like viruses. Thanks for watching.